Good afternoon, good morning, or good evening, depending on where you're joining us from today. And we welcome you to our industry partner update with Dyne Therapeutics. My name is Rick Moss, and I'm the Director of Strategic Partnerships at Jesse's Journey. To kick things off, we're going to ask Rochelle to conduct a couple of polls. First of all, we want to know who's on the webinar today. You'll see a few questions pop up on your screen, and we'll encourage you to share your response and then get the results. Okay, Rochelle, have we got some results from poll one?
Yeah, so it looks like the majority of people are joining us from Ontario and Quebec, but a few from the Prairie provinces as well, and same with the Atlantic on the East Coast, and a few people outside of Canada.
Awesome. And we have just one or two more polls we'd like to go through. We would like to know where you are. Are you new to Jesse's Journey? Have you done anything with us before? Is this your first event that perhaps you've attended? And so if you could just answer that poll question for us, we'll get the results momentarily. Thank you.
And again, it looks like everyone has attended a Jesse's Journey event in the past, which is fantastic.
Great.
People that already know all about us.
Great. And last question, last poll is, what is your connection to Jesse's Journey?
Okay, so as the results are coming in here, we have a couple of parents and caregivers of children with Duchenne or Becker muscular dystrophy. We have those living with Duchenne or Becker muscular dystrophy online with us today, a few extended family and friends of children with Duchenne or Becker, researcher. So all across the board, I'll end the poll and share those results.
Okay, thank you. Well, for those of you who may not be totally familiar with Jesse's Journey, our organization began when the Davidson family from London, Ontario received the news that their middle son, Jesse, was diagnosed with Duchenne. That was back in 1986. They quickly realized how little was being done to find solutions for this devastating disease. So John and Jesse set out to raise awareness for this rare disease and funds for research. In 1995, John pushed Jesse in his wheelchair across Ontario, some 3,339 kilometers. And then a few years later, John walked across Canada, 8,300 kilometers. These two incredible feats launched Jesse's Journey, Canada's only registered charity dedicated to defeating Duchenne. More than 25 years later, here we are. We've become the country's largest funder of Duchenne research, investing close to $15 million in projects around the world. And we've attempted to unite the Duchenne community to advocate for access to treatments in Canada. We also provide education and support programs for families living with Duchenne and healthcare professionals through conferences, our family forums, our webinars like today, as we hear the latest research from Dyne Therapeutics. And now it is my pleasure to introduce you to Dr. Dugar, Senior Vice President of Global Medical Affairs at Dyne Therapeutics. He began his career at the National Institutes of Health, and prior to joining Dyne, he served as Vice President and Global Head of Medical Affairs at Sarepta Therapeutics. Today, Dr. Dugar will introduce us to the Dyne Therapeutics' latest research and clinical data from their FORCE platform, which drives their efforts to develop targeted therapeutics with the potential to be life-transforming for patients with serious muscle diseases like Duchenne. I would also like to extend a thank you to Molly White, the Vice President and Global Head of Patient Advocacy and Engagement, who was responsible in helping us pull this webinar together. We ask that you please put any questions you have into the chat throughout the webinar, and we will have a few minutes at the end to answer those. And we also have questions that families have sent who can't attend today. And now, without any further ado, it is my distinct pleasure to turn things over to Dr. Dugar.
Wonderful. Thank you, Rick. Thank you, Rochelle. I am going to attempt to share my screen so that we can get started. Please bear with me. All right. All right, there we go. First of all, forgive me, I have my camera here, but I have a screen to my left that I'm looking at, so hopefully that's not distracting. First, I wanted to thank Rick, Rochelle, and just everyone at Jesse's Journey for inviting me and Molly to this talk today. It's a true privilege to speak to all of you today, and the journey that Jesse's Journey has been on has been extraordinary. And I've known the organization for several years now, and the work that has been done for the community is truly amazing. So I'm really honored to be here today to be able to provide some information on what we're doing at Dyne Therapeutics right now. So today I'm going to talk about what we call the FORCE platform. It's the scientific platform that we are using to develop potential therapeutics for Duchenne muscular dystrophy. I'm going to be talking about the newest data from our Duchenne program and then touch on our clinical development plans. So these are our forward-looking statements. I definitely have to show them, and it just states that I'll be making statements that involve risks and uncertainties. So at Dyne, it all starts with our mission. We're laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne muscular dystrophy being a priority. Our mission is truly core to who we are and what we do every day. And I've been at the company for about 8 months, and it truly is a company that has the patients at the heart of everything we do. We've created, thanks in large part to Molly, a multifaceted program involving community webinars, conference participation, fireside chats, and other outreach to raise visibility and gather input to advance our drug development efforts. And over the past few years, we've been validating our platform and technology, including our ability to target delivery of potential therapeutics to muscle, and have generated what we think is very exciting preclinical data across multiple indications and disease models. And our focus today is on sharing our newest data from the DMD program. So we're building Dyne as the leading muscle disease company to deliver for families living with Duchenne and other muscle diseases. On this slide, we show our 4 core strengths to help us deliver on that promise. First, we intentionally designed our FORCE platform to overcome the current limitations of muscle tissue delivery of modern oligonucleotides. And I'll talk much more about this in a few slides. The goal for our DMD program is to develop a therapy that will robustly deliver our exon skipping therapy to cardiac and skeletal muscle, including diaphragm, hopefully leading to substantial increases in dystrophin expression with relatively infrequent dosing to modify disease progression. Second, it's our focus on rare muscle diseases. As I mentioned, we've generated very exciting preclinical data that shows the power of our technology across multiple diseases and in DMD in particular. And I'm pleased to highlight some of that data in this presentation. The third, it's really our clinical plans. We are laser focused on moving into the clinic, and I'll provide some more details on that in a few slides. And then finally, I would emphasize that we have exceptional in-house and advisory expertise that has deep knowledge of Duchenne with a world-class scientific advisory board, which includes luminaries such as Dr. Francesco Montoni and Lou Kunkel, whom you know well. So this slide provides an overview of Duchenne, which you all know very well. Really, what I want to communicate here is that our goal is to develop a best-in-class targeted exon skipping treatment that substantially improves dystrophin restoration, where it enables much less frequent dosing than what occurs with current agents, and with the hope of potentially stopping or reversing disease progression. So this slide describes our FORCE platform. Our FORCE platform consists of a specific delivery mechanism called a Fab, or an antigen binding fragment, that we've engineered to bind to a receptor called TFR1, or transferrin 1 receptor. This receptor is found across the surface of muscle cells. It's highly expressed across these muscle cells, and that includes cardiac muscle, skeletal muscle, and smooth muscle. Now, if you look at the Y-shaped figure on the left, that's how a full antibody looks like. Our FORCE platform uses the top piece, or the Fab, the antigen binding fragment, which binds very tightly to the TFR1 receptor on muscle cell walls. We believe Fabs have several advantages over these full antibodies or these monoclonal antibodies, including their much smaller size, which helps with distribution to muscle tissue. And again, I'll remind you that that is one of the biggest issues with current agents, the ability to get enough of the PMO, enough of the exon skipping drug into the muscle cells. And so, this Fab actually is quite a smaller size than a mAb and allows for that enhanced distribution. So what does the Fab do? Well, the Fab acts as a kind of key that unlocks that transferrin 1 or TFR1 receptor and allows our therapeutic oligonucleotide to enter the skeletal, cardiac, and smooth muscles to target the genetic cause of disease at the level of the nucleus. So we want to drive in this case, our PMO into the nucleus so that it can actually do the job it needs to do. Now we link our Fab to the potential therapy with a linker, which you see at the bottom of the slide. And this linker is a clinically validated element. And what I mean by that is it's been used in products that are on the market. So we really understand this linker very well. And it enables precise connection of the PMO to the Fab. Well, historically, as I've said a few times now, the challenge is the inability to get enough of these PMOs into the muscle cells. And as you can see on the right-hand side, we are using a PMO. They have been shown to be able to demonstrate exon skipping and provide some benefits in terms of dystrophin production. And so our technology links the antibody to the PMO to be able to drive the payload or this oligonucleotide into the muscle cells. And we've developed a short video to illustrate our FORCE platform, and I'm going to try to play it now for you. I'm going to have to stop sharing. Okay.
At Dyne, we are focused on advancing life-transforming therapies for people living with serious muscle diseases. We are utilizing our FORCE platform to develop modern oligonucleotide therapies that are designed to overcome limitations in delivery to muscle tissue. Our therapeutic candidates consist of three components: an antigen-binding fragment antibody, or Fab, a clinically validated linker, and an oligonucleotide payload. The Fab is conjugated or connected to the therapeutic payload using a linker. Our proprietary Fabs are engineered to bind to the transferrin 1 receptor, or TFR1, which is highly expressed on the surface of muscle cells. The Fab engages with the TFR1 to facilitate the delivery of the payload into the muscle, where it aims to target the genetic basis of disease. Dyne is advancing therapeutic candidates for myotonic dystrophy type 1, Duchenne muscular dystrophy, and facioscapulohumeral muscular dystrophy. The modularity of the FORCE platform enables the selection of different types of payloads to match the genetic basis of the disease, including phosphorodiamidate morpholino oligomers, or PMOs, to promote nuclear exon skipping and DMD, and small interfering RNA for targets in the cytoplasm. With the FORCE platform, Dyne is aiming to deliver therapies with the potential to transform the futures of people living with serious muscle diseases.
All right, hopefully that played and everybody could hear it well. So now I'm really excited to present some of the newest data that we've shown recently at the Muscle Study Group Annual Scientific Congress, and also our recent R&D Day that we held last week. As an aside, all the information, including the slides and the webcast replay from our R&D Day, are available on our website in case you're interested in much more information than what I'm able to show in the time we have here today. So on this slide, we show our force platform in the MDX model. It's a standard animal model in Duchenne. Now, these MDX mice have a mutation in exon 23. So you can see force resulted in robust and durable exon skipping, which is shown on the top half of the slide, and dystrophin production, which is shown on the bottom half of the slide, across the quadriceps, diaphragm, and heart. Now, the profound exon skipping is shown as early as week 1, and you see the highest levels of dystrophin production at week 4, but extending out to even 12 weeks. We were able to achieve very high levels of exon skipping in the quadriceps, diaphragm, and heart, where even at week 1, we see over 40% skipping in the quadriceps, and heart over 30, and then over 30% in the diaphragm. Now, on the bottom half of the slide, I'll just take you through the dystrophin data in each muscle. So it stands to reason that if you're able to show robust exon skipping, then dystrophin production should follow. So indeed, at 30 milligrams of the forced compound, you can see over 40% dystrophin restoration at 8 weeks in the quadriceps, and about 90% in the diaphragm, and 78% in the heart, and both of those at 4 weeks. So we're extremely excited to see these levels of dystrophin restoration, especially in the diaphragm and heart, where current PMOs have had significant limitations. So on this slide, on the top panel, we show dystrophin localization in the muscle membrane, or the sarcolemma, after a single dose of 30 milligrams per kilogram. So this localization is important to see to provide confidence that we would see some functional benefit. At 8 weeks, we see profound results, including 83% dystrophin positive fibers in the quadriceps, 80% in the diaphragm and heart. And what's really interesting as well is on the bottom half, you can see this red staining, and it's really showing you the tissue architecture. So one can see restoration of the cellular architecture as well. And when we've shared these with thought leaders, these data with thought leaders, they've shared our excitement in the level of dystrophin production and in the architecture restoration. Next, in non-human primates, which is another important step in generating preclinical data before going into the clinic, we evaluated Dyne 251, which is our lead candidate going into the clinic. And we've done this with multiple dose regimens, as you see here. So I'll just kind of orient you to the slide. So first of all, what you're seeing is a study here where we dosed these NHPs with 2 doses of 30 milligrams 2 weeks apart, and then evaluated their tissue 4 weeks at 4 weeks. Another arm in which the NHPs received weekly 30 milligram per kilogram doses, and then evaluated their tissue at 4 weeks. And then another arm in which we gave them 5 weekly doses of 30 milligrams per kilogram and evaluated their tissue 8 weeks later. As a reminder, these are healthy animals. So you cannot look at dystrophin restoration in this model. We can only look at exon skipping, but because they're healthy, there's no way to evaluate dystrophin restoration. So the bottom line here is that with our platform, time on therapy matters. The 2 and 4 doses at 4 weeks are comparable in terms of the level of exon skipping, which you can see with the light blue and the medium blue bars. And you can see across all of the tissues, it's relatively comparable in terms of exon skipping. But when you look out to 8 weeks, we observe potentially transformative levels of exon 51 skipping across the muscles. 18% in the quadriceps, 52 in the diaphragm, and 43 in the heart. And again, I would emphasize how amazing that exon skipping is, especially in the heart and diaphragm, which are muscles that obviously weaken significantly over time, leading to many of the cardiopulmonary issues that are part of the Duchenne disease progression. So we also evaluated Dyne 251 in a 5-week toxicology study. Again, another standard study in which you want to understand how well your candidate or how well your drug is tolerated. And what we saw was a favorable safety profile in this GLP toxicology study, with no dose-limiting toxicity observed after those 5 weekly doses up to a maximum feasible dose. Importantly, we pushed the dose, dosing many folds higher than the 30 milligrams per kilogram in this study. And so we're really pleased with the safety profile as we advance into the clinic. I'm going to switch gears and now start to talk about our plans to get into the clinic. So as we prepare to launch our first clinical development program, we feel we've done quite a bit of work to get the best advice possible as we develop our plans. We've engaged with advocacy groups, patients, and key thought leaders from various disciplines, including pediatric neurology, cardiology, pulmonology, and physical therapy, just to name a few. Importantly, we've gathered this feedback across the globe. So we've gotten feedback from opinion leaders both in the United States and outside of the United States, with a great deal of experience running early and late stage clinical trials. And it's been incredible that they've shared their thoughts and advice on our overall clinical development program. So we've asked them we've asked them for feedback on many elements, including our clinical trial design, the patient population, our biomarkers that we should be looking at, the functional endpoints, and then of course, key safety considerations. They've also been very generous in helping us pull learnings from natural history studies, which is absolutely critical. And it's clear that they believe dystrophin continues to be a key endpoint for this disease. And as you can see on the right side of the slide, we've also received advice from global advocacy leaders, patients, and caregivers that have provided insights based on their living with and being the voice of the Duchenne community, as well as their experience in past and current clinical trials. So this input's been extremely critical in ensuring a patient-centric design, and the advice they provided includes key considerations for the patients and families in terms of choosing a clinical trial, because there are so many options out there, which is wonderful. Feedback on their thoughts related to biopsies or a placebo arm, functional outcomes that we're proposing for both ambulant patients and non-ambulant patients. And then also being very clear with us on what the most burdensome or difficult parts of the trial might be, and then how to ensure patients and families are supported throughout the process. So based on feedback we received from thought leaders, patient groups, and regulators, we've developed the proposed clinical study for Dyne 251 in patients amenable to exon 51 skipping. And I'm going to walk you through the key elements here. So our clinical trial for Dyne 251 will be a global, multiple ascending dose, placebo-controlled study with a long-term extension. We plan to enroll patients with symptomatic DMD and mutations amenable to exon 51 skipping. Our goal is to enroll about 30 to 40 participants, aged 4 to 16 years, and who are either ambulant or non-ambulant. So we really do want to be mindful of putting a program forth that can serve, every stage of the disease. Some of the endpoints we plan to evaluate include dystrophin by Western blot, measures of muscle function, both lower limb function, upper limb function, as well as pulmonary function. And then, of course, importantly, we want to evaluate safety and tolerability throughout the study. We plan to submit our IND this year, And so we're really excited for, I'm sure many of you know, that's the next step prior, that's the step that we have to get through prior to getting into the clinic. So everything is quite on track, and we're excited to announce that we'll be submitting the IND in the fourth quarter of this year. And more details will be available closer to when we initiate the trial. So while our initial focus is on our exon 51 program, as I mentioned earlier, our goal is to build a best-in-class pipeline to address the 80% of patients with Duchenne amenable to exon skipping. We'll be looking to rapidly advance programs in exons 53, 45, and 44. And the data I've reviewed, combined with the deep engagement we've had with multiple stakeholders, we believe has positioned us well to make this very exciting transition to the clinic. I'm looking forward to the important work ahead as we advance our efforts to develop new potential therapeutics for patients living with Duchenne. So this slide importantly recognizes just a few of the very many voices and comments that we hear. We're committed to understanding the community of people living with muscle diseases and Duchenne in particular, and ensuring that your perspectives and experiences are included in our drug development program and in everything we do moving forward. This is central to our work each and every day. So thank you so much for advising and working with us. And finally, on this slide, you can see Ravi, Beth, and Diana and their families, who are all part of the Dyne family. At Dyne, we make decisions with patients and families in mind, and this is absolutely what drives us each and every day. Thank you.